Welcome to Biotechnology. Biotechnology is the manipulation of organisms or their genetic components to make useful products such as sugar. Genetic engineering is the direct manipulation of genes for practical purposes. Genetic engineering can be applied to any organism from a virus to a sheep. For example, genetic engineering can be used to produce plants to have a higher nutritional value or can tolerate exposure to herbicides. To work directly with specific genes, scientists prepare gene-sized pieces of DNA in identical copies, a process called DNA cloning. Most methods for cloning pieces of DNA in the lab share general features such as the use of bacteria and their plasmids. DNA cloning results in recombinant DNA that has been formed artificially by combining constituents from different organisms. Gene cloning uses bacteria to make multiple copies of a gene. Foreign DNA is inserted into plasmid and the recombinant plasmid is inserted into a bacterial cell. The recombinant bacteria is a microorganism whose genetic makeup has been altered by deliberate introduction of new genetic elements. Reproduction in the bacterial cell results in cloning of plasmid, including the foreign DNA. This results in the production of multiple copies of a single gene. Copies of this gene can be inserted into a plant and used for pest resistance. Restriction enzymes cut DNA molecules at a specific DNA sequence called restriction sites. Restriction enzymes cut sugar phosphate backbones. DNA fragment added from another molecule is cut by the same enzyme. Base pairing occurs. DNA ligase then seals the strands. DNA ligase is an enzyme that seals the bonds between restriction fragments. PCR is a laboratory technique that is used to copy a specific region of DNA. It can produce many copies of a specific target segment of DNA. For example, if there is a piece of hair at a crime scene, it will go through a 3-step cycle which brings about a chain reaction that produces a population of identical DNA molecules. In the first step, denaturation, heat is used to denature the DNA strands. This provides a single-stranded template for the next step. In the next step is primer annealing. The reaction is cooled down so the primers can bind to their complementary sequence on the single-stranded template DNA. After is primer extension. The reaction temperatures are raised so TAQ polymerase extends the primers, synthesizing new strands of DNA. This process uses a gel as a molecular sieve to separate nucleic acids or proteins by size. It produces one or more organisms genetically identical to the parent. Stem cell can reproduce itself indefinitely and differentiate into specialized cells of one or more types. Stem cells isolated from early embryos are called embryonic stem cells. They are able to differentiate into all types. There are also adult stem cells which replace non-reproducing specialized cells. Example of synthesis of small molecules for use as drugs is insulin. GMOs contain genes from DNA of one species that are extracted and inserted into the genes of a plant or animal. When you walk into the grocery store, you are surrounded by genetically modified foods. More than 70% of packaged foods in North America contain GMOs.